Hello guys, I am Shubham from Tutorials. This video is about site-specific recombination. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So what is site-specific recombination? It is nothing. We have previously discussed about recombination, homologous recombination. This is recombination only, but that occurs at specific sites. Okay, that occurs at the specific sites. So recombination occurring at specific site is known as site specific recombination. So what are the specific sites? Specific sites are known as recombination sites. And the second thing is the recombinase. Okay, important enzyme recombinase. Right? Recombinase enzyme. Then let's talk more detail about those things now. Recombination sites have two components basically. First is recombination recognition sequence. Recombinase recognition sequence. I will write here full form also. Recombinase recognition sequence. This is the first component. And the second component is crossover region. Okay, where the crossovers take place. Recombinase enzyme is also of two types. First is serine recombinase. Serine recombinase. And second is tyrosine recombinase. Tyrosine recombinase. Now let's see what is it exactly. I will draw a DNA here. Okay, so this is a DNA. Alright, now DNA has a sequence. This is a sequence from here to here. Okay, all this region completely consists of a recombination site. A recombination site. Alright, within this region there are sections. First of all, let's see. I start from here to here. Okay, same here. Here also. From here to here. Okay, let's make a marking there. So these are recombinase recognition sequence. Recombinase recognition sequence. Okay, and in the middle, this thing is a crossover region. Crossover. Alright. Now what is this recombinase recognition sequence? This is a sequence present in the DNA where a recombinase enzyme, it may be serine or tyrosine, will come and bind. Okay, enzyme will come and bind here. Which enzyme? A recombinase enzyme. So this is a recombinase enzyme which will recognize this sequence in the DNA and will bind to it and start the process of site specific recombination. right? Then this recognition sequence is having a symmetric sequence, symmetrical sequence. We can see symmetry, and this crossover region is asymmetry. Asymmetrical sequence, basically known as asymmetry. So how this asymmetry is generated? Because the repeats are no palindromes. I hope everybody knows this palindrome. Palindrome sequence is what, suppose we go on reading from this sequence. The sequence is same from right to left as well as left to right. Means both the directions we can see same sequences. Right? No palindrome. No palindrome means the repeats are not palindrome, and this will give rise to asymmetry. Right? So this crossover region is having asymmetry, and recombinase recognition sequence is having a symmetry. Right? So this is a diagram. We can see here site specific recombination is giving rise to inversion. You can see here A is there, here B is there, and here some part of blue is come here. So inversion is taking place. Okay, so what is happening? From this goes this side, A goes this side, B come this side. So this is giving rise to inversion. Right? Then we can see here this is an example of integration of lambda phase genome. You can write here integration of lambda genome into bacteria. So this is the example of site specific recombination. So we can see here there is a specific site in the phage as well as bacteria. See, this is the site, dark red and dark blue. The exchange is taking place between these two sites only, specific sites, and then the whole genome is getting inserted. See, the bacteria within those specific sites only. Right? Then again, the recombination takes place in three types. Okay, three things can happen. This is a DNA one. What happens now? AB has to get integrated, so it gets inserted. See, insertion. Right? This is a insertion. Okay, insertion. We can see our crosses thing. So A, besides A, there will be B, Y, like this, and X with B. Okay, so this is the insertion. Now again, see, same DNA is there. After insertion, it will get out also. Okay, means we can say excision. This second image is of excision, we can say, means the integrated genome or DNA is coming out. So this is deletion. Formation of this thing happen, and here will be the cross. Right? See, again X will combine with Y, A will combine with, so they are separate now. In the third step, we can see inversion. Inversion, right? Inversion forms a coil like thing, this coil. Okay. See the arrows, basically. Arrows this side only. Thus this type of coiling has taken place. As well as here. Here they are facing towards each other. So here inversion will take place. Right? Within the same DNA. Then this I have shown you just now only. Recombinase sites. Okay. Recombinase sites have two sequences: recombination, recognition, and crossover region. Right? See, recombinase binds to recognition sequence. This is a recombinase. This is a recognition sequence. Same here with this another DNA. And then the recombination will take place. Recombination means exchange of strands. Just okay. Then this is a reaction slide. How reaction takes place. This is very important. Reaction of recombinase. What happens? It may be serine or tyrosine. Anything. See carefully. This is a rec enzyme. Recombinase with serine. Serine having OH group. This OH is attacking on this phosphate in the DNA. Right? Then what happens? The bond has been broken down. See. Okay. So we can see a complex of this thing in this picture. Rec is combined with this phosphate. This bond has been broken down. But again what happens? A rejoining takes place. Rejoining. Again this OH will attack phosphate. Rejoining, we can say resealing as well. So the rec serine again is free. Okay. What we can understand from this picture is that bond is broken as well as resealed. Very important feature of site-specific recombination. Bond is broken as well as resealed. This serine recombinase or tyrosine recombinase, what they do? They break the bond as well as resealing also takes place. So thus, this process is known as conserved site-specific recombination. Conserved site-specific recombination. Conserved means the bonds are conserved, right? Sequences are conserved. Sequences are not lost. Same like homologous recombination. But here the enzymes are different. Serine recombinases are there. Tyrosine recombinases are there. Same here, we can see the proteins. Proteins known as sp11, which cause a break, introduce a double strand break in the DNA for start of homologous recombination step, right? So this enzyme also acts in the same manner as well as DNA topoisomerases. These enzymes also act in the same manner. Okay, so this site-specific recombination is also known as conserved site-specific recombination because we can see the sequences are conserved, the bonds are conserved, original bonds. Okay, original bonds are conserved. Serine recombinase just attacks the phosphate. Again, the OH of the DNA will attack the phosphate. So ultimately, serine recombinase is free only, right? So we can say it is like a recycling thing. Now this is the example of the recombinase enzyme. Very important. Salmonella in invertase. What is the function? Inverts a chromosomal region to flip a gene promoter by recognition peak sites. Allows expression of two distinct surface antigens. Okay. Transposon, this is related to transposition basically. But remember this is an example of serine family. What it does, promotes a DNA deletion. Transposon deletion reaction takes place to resolve the DNA fusion that results from replicative transposition. We will see this in the separate video. What is replicative transposition? Recombination sites are called rest sites here. Then tyrosine family, bacteriophage lambda integrase, very important, promotes DNA integration. We know that lambda phage genome integrates Okay, lysogenic cycle. Excision also takes place of this lambda genome into and out of specific sequence on the E. coli chromosome, right? And here recombination sites are called attachment sites, right? Then the second enzyme, phage P1, what it does, promote circularization of the phage DNA during infection by recognition sites called lock sites on the phage DNA, right? Then the next is Escherichia coli, XCRC and XCRD. It results in DNA deletion reactions that convert dimeric circular DNA molecule into monomers, recognizes both plasmid bond sites, CER, and chromosomal sites. And the next is East FLP. Inversion takes place of yeast to microplasmid to allow for DNA amplification reaction called rolling circle replication. So this is also very important. A rolling circle replication takes place because of this yeast FLP, and the recombination sites are called FRT sites. Okay, right? So let's see now individually how this enzyme acts. First of all, serine recombinase. Serine recombinase. Nothing. I will just write down in short. In this serine recombinase, what does? All the 4 strands are glued at once. Okay, all the 4 strands are glued at once. See how. Here is this is the first pair of DNA, this is the second. Alright. Here comes enzyme 1, R1, R2. Here R3, R4 on red one, R1, R2 on blue one. Now what it does, it will attack the phosphate. So 3' OH is free now, as well as R2. It is attacking this one 5'. Then this 3' is free. Same here with R3 and R4 phosphate. 3' OH is free. 3' OH is free. It is attacking on 5' phosphate. All the 4 strands are cleaved at once, right? This only we have to remember. Swap DNA partners means what? Exchange of strands. Just exchanging the strand takes place and then rejoining. That's only the case here, right? That's only the case here. Second thing about tyrosine. In tyrosine, what happens? 2 strands are broken first. 2 strands are cleaved first, and then other 2. And then other 2. We can see formation of holiday junction here. Okay, do note. Holiday junction formation is seen in the tyrosine case. Let's see. See, here R1, R2, R3, R4. First of all, top strand cleavage means this strand is cleaved first from the first pair and from the second pair. Top strands are cleaved. Okay, so this R1 and R2, R3, R1 and R3 are cleaving the strands. We can see here. Okay, here 5' OH is there. Here 3. Here also 5' OH. So this will become the 3', right? Now this is going to attack this one, R1, right? And this is going to attack this one, R3. Okay, so thus we can see a holiday junction formation. R1 with this R3. So here we can see formation of holiday junction. After this thing happens, there is a cleavage of bottom strands. Cleavage of bottom strands. Then the bottom strands are cleaved and the exchange will take place. to finish recombination. That's it. So these are only the points we have to remember in this site-specific recombination. Very easy. I hope this video helps you. If you find this video helpful, do let us know in the comment section.